Yup, that's us. You're probably wondering how we got into this situation. Well, let us tell you.
Currently, 10 million people in the world suffer from inflammatory bowel diseases, or IBDs. Inflammatory bowel disease is an umbrella term mainly referring to Crohn's disease and ulcerative colitis. These diseases have similar symptoms but differ in their pathology. Crohn's disease can manifest in all parts of the digestive tract and usually appears in patches, while ulcerative colitis only affects the large intestine, usually in one continuous patch. Both of these diseases are autoimmune diseases where your body attacks itself. The exact cause of IBD is unclear. It usually starts due to immune dysregulation, but genetics, diet, stress, age, and environmental factors also play a role in its manifestation. The problem with IBDs is that they are usually diagnosed after the first symptoms appear, which usually means that the patient has already experienced permanent inflammatory damage to the digestive system, which can even lead to whole parts of the gut having to be removed. The disease is chronic and comes in symptom flares, usually with abdominal pain, diarrhea, rectal bleeding, fatigue, and appetite loss, and with this, often a sudden weight loss. All this leads to severe reduction in quality of life for the patients. However, recent studies have connected the dysregulation in the gut microbiome with the emergence or exasperation of IBDs. The gut microbiome consists of all bacteria, fungi, and viruses that live in our gut, most of which are beneficial to us. They help digest our food, make nutrients more bioavailable to us, help regulate the barrier of the gut towards pathogenic organisms, and even actively train and regulate our immune system. Some bacteria, mainly of the Firmicutes and Bacteroides species, produce small molecules called short-chain fatty acids, which are small carboxylic acids only 2 to 6 carbon atoms long. They are produced during the fermentation of complex starches and dietary fiber in the gut. These short-chain fatty acids act as energy source for the bacteria, but also as signal molecule for the host cells. The most common short-chain fatty acids are acetate, propionate, and butyrate. They appear in specific ratios in the stool and can tell us about the gut health of a person. These short-chain fatty acid ratios are different in patients with IBD. Gut dysbiosis usually precedes the change in short-chain fatty acid ratio, which itself precedes the disease manifestation. So if we could find a way to reliably quantify short-chain fatty acid ratios in stool samples in an accessible and cheap way, IBDs could be diagnosed earlier, which could prevent permanent inflammatory damage to the gut.
To address this issue, our goal is to develop an early detection system that would enable people to check for patterns in dysbiosis linked to IBDs in order to discover autoimmune inflammation early. So before we could start, we talked about our plan to various experienced people, but the response was always the same. Short-chain fatty acids are 2 small molecules and vary in only 1 carbon atom to specifically detect and distinguish them from each other using tools other than mass spectrometry. But we continued and came up with the idea that is less complex and cheaper than that, our GLOW.coli. GLOW.coli is an engineered E. coli strain which carries a simple genetic reporter construct. The constructs consist of specific promoters that activate upon direct binding by short-chain fatty acids, or in one case by a metabolic derivative of the responsive short-chain fatty acid. So when GLOW.coli encounters its corresponding short-chain fatty acids, it triggers the reporter to produce a detectable signal. For the acetate-inducible system, the GLIN-AP2 promoter is used, which is normally induced by the nitrogen regulator 2 under nitrogen deficiency. The PSHA promoter is used for the butyrate-induced construct. Butyrate thereby forms a complex with the native E. coli leucine-responsive regulatory protein and can thus bind to the promoter of the PSHA gene. And lastly, in order to create a propionate-inducible expression system, we use the PRPB promoter, which is responsible for the expression of the propionate catabolism genes. They additionally encode PRPR, which acts as a positive regulator of this promoter. In E. coli, propionate is converted to 2-methyl citrate in 2 consecutive steps. In the presence of 2-methyl citrate, PRPR initiates transcription of the catabolic genes by binding to an enhancer-like element located at the distance 5' to the PRPB promoter and contacting the RNA polymerase by DNA loop formation. In the end, by comparing the fluorescent intensity induced by the short-chain fatty acids under healthy conditions with ratios commonly seen in IBD patients, this can be used to measure the concentration of that specific short-chain fatty acid, and by taking all short-chain fatty acid concentration together to make a statement about a dysbalance ratio. To transform our competent bacteria with our reporter constructs and create our GLOW.coli, we agreed to perform a Gibson assembly. We took advantage of the fact that Gibson assembly requires overhangs that are complementary to the vector by inserting one Him3 restriction site forward and one reverse into the extension of the sequences. These do not interfere with the coding sequences, but allow the inserts to be accurately excised from the plasmid, provided it has been correctly inserted into the plasmid. So after linearization of our plasmid, which we confirmed using agarose gel electrophoresis, we used the linearized vector for the Gibson assembly. Afterwards, we transformed the bacteria, and prior to streaking the transformed bacteria on the plates, we spread X-GAL on the LB agar plates to perform a blue-white selection. Since our plasmid contains the gene of beta-galactosidase at the position where our promoter and reporter genes are inserted, the beta-galactosidase becomes inactivated, causing the colonies to remain white, while the E. coli colonies without the inserts appear blue. With our white colonies, we conducted a colony PCR to confirm that our Gibson assembly worked. By that, we were able to show that our inserts were ligated at the correct site. Here I also want to mention that in our first attempt, we struggled to perform a Gibson assembly, since we could not show the expected bands by performing colony PCR, nor could we excise the inserts using our additional Him3 restriction sites. Keeping this attempt in mind, our aim was to investigate whether any difference in fluorescence could be observed when exposing the same number of bacteria to different concentration of short-chain fatty acids. To accomplish this, we utilized all 9 constructs, meaning acetate, butyrate, and propionate, each with GFP, BFP, and TEC-RFP. The concentrations of short-chain fatty acids used were 1000 to 0.01 fecal equivalent with a 1 to 10 serial dilution, where we calculated the mass of the specific short-chain fatty acids in 0.01 gram to 1000 grams of mouse feces. So the total mass of a specific short-chain fatty acid that is present in 1 gram of mouse feces corresponds to 1 fecal equivalent, which then can be scaled up or down. As a control, we use the autofluorescence of the bacteria without stimulation to confirm the effect of short-chain fatty acids. It should be noted here that the concentrations greater than 10 to 100 fecal equivalent exceed the concentrations of short-chain fatty acids that are realistic to achieve in an experimental setup. However, it was interesting to observe the impact of short-chain fatty acids on bacteria when exceeding physiological concentration. Interestingly, the fluorescence increased with higher concentration. Also, it is observed that the fluorescence peak decreases over time, especially at higher concentration. Therefore, we conducted a literature review and found that certain substances can alter the morphology of bacteria, for example by damaging the cells and thereby increasing autofluorescence. Prokaryotic cells such as E. coli can exhibit an intrinsic fluorescence due to the presence of fluorescent cellular structural components and metabolites. Examples for these compounds are flavins and NAD, which are responsible for the majority of cytoplasmic autofluorescence. Both the fluorescence of FAD and NAD are redox dependent, meaning that while the oxidized form of FAD exhibits a strong fluorescence, the reduced form is less fluorescent. For NAD, it is the other way around. Generally, the autofluorescence depends on the metabolic state of the cells as well as on different stressors that affect the cells, for example, the use of antibiotics. Accordingly, we will perform experiments in the future where we will try to control the metabolic state of our GLOW.coli. Another factor that we did not consider in a wet lab is the endogenous acetate a cell is exposed to all the time. For that, our dry lab had an interesting idea.
In dry lab, we quickly figured out that we might encounter a problem with our bacterial reporter system influencing the short-chain fatty acid concentration in the sample. Metabolic processes taking place in the E. coli would distort our measurement when, for example, acetate is produced or consumed by the bacteria. However, it would have been far too complicated to investigate metabolic interactions in the wet lab. That is why we decided to build a stoichiometric computer model of E. coli to figure out what is going on inside the cells. The model was created by taking chemical reactions and translating them into differential equations describing the individual reaction rates. The equations were then composed into a stoichiometric matrix representing the entire metabolic network. We decided to model a 2-component system containing the bacterial cell and the medium. This enabled us to analyze how short-chain fatty acids enter and leave the cell. We performed metabolic control analysis on our model and received a set of elasticity coefficients. The values highlighted in green indicate which metabolite concentrations contribute most to the values of the reaction rates. Finally, we ran a time course simulation to find out how E. coli influences extracellular acetate concentrations over time. To calibrate the simulation, we used proteomic data of bacteria using glucose as energy source. As you can see in the resulting diagram, the red line of extracellular acetate concentration is little affected by E. coli. That means that acetate is not metabolized in the presence of glucose. Consequently, we decided to run our measurement in glucose medium in order to not confound the short-chain fatty acid concentration of the sample. We hope that our model can also serve as a starting point for other iGEM teams for developing solutions for their own metabolic models. Of course, we and all other sub-teams of our iGEM team conducted several interviews with experts as well as many other human practices collaborations.
For the human practice part of our project, we conducted several expert interviews. At first, we interviewed Professor Mario Zeiss and brainstormed about short-chain fatty acid detection using bacteria as well as short-chain fatty acid investigation using a gut-on-a-chip device to replicate the intestinal environment. In a second interview, Professor Zeiss told us about the impact of IBD, its pathology and dysbiosis. Additionally, we also interviewed Professor Andreas Bukowski about the possibilities of modeling the central metabolism of E. coli. Here we were informed about the obstacles that come with this project and we were provided with many resources on how to proceed. Lastly, we interviewed Professor Sonnewald to discuss the use of a metabolic model of the endogenous acetate pathway in E. coli to simulate its interaction with our biosensor construct. In addition to the interviews, we also developed a questionnaire to investigate how widespread the knowledge about the important role of nutrition for the immune system is in society. For this, we developed a questionnaire that consisted out of 11 questions about eating habits, nutrition, and its influence on overall health. The questions were provided in English, German, and Spanish. The results showed that the participants were between 18 and 65 years old, and that a general awareness about the effect of antibiotics on the microbiome, as well as the impact of nutrition on the immune system, was present. More than 60% of participants have a healthy diet, and many supplement their diet by using supplements such as magnesium, calcium, and iron. The meat intake varies widely among the questioned group of people. While some people eat meat more than 3 times a week, there are people that only eat meat once a week, and some are even vegetarian. Interestingly, the highest number of gut-related diseases was found among the Spanish-speaking participants. For the education part of our project, we developed an accessible educational concept for students from ages 11 till 18 about gut health, immunology, and a healthy diet. Furthermore, this also creates resources in the form of games for teachers to use in the future. Besides the school project, we also created educational infographics for Instagram to reach as many people as possible and to inform them about IBDs and their treatments, as well as the gut microbiomes, short-chain fatty acids, and also our project. By doing this, we provide them with the information in a compact and easy to understand manner. This ensures that our project and the scientific background are accessible for people independent from their previous knowledge. Lastly, we also created a podcast. Here we wanted to introduce the concept of iGEM, past teams and projects to a wide range of listeners in a contemporary way. This also was used to educate the audience about challenges of high relevance for society and their potential solutions.
Our GLOW.coli would solve the problem of late diagnosis for IBD patients and would lead to an improved quality of life.
We developed a cheaper, simpler and more accessible alternative to conventional methods that could potentially alleviate strain on the public health system.